Good evening, everyone, and welcome to the Real Science Exchange, the pubcast where leading scientists and industry professionals meet over a few drinks to discuss the latest ideas and trends in animal nutrition. Hi, I'm Scott Sorrell, and as always, I'm joined by the incomparable Dr. Clay Zimmerman. Welcome back, Clay.
Thanks, Scott. Good to see you again.
Yeah, always good to see you. Tonight, we continue our mini-series of lectures presented at the 2022 Tri-State Nutrition Conference titled Exploring In Utero Influences on Transgenerational Performance. Last week, we featured Dr. Jack Britt and his presentation titled Epigenetics Will Change How We Manage Cattle. If you missed that one, you'll want to go back and listen to it. This week, we're highlighting Dr. Jimena Laporta from the University of Wisconsin and her presentation titled Phenotypic and Molecular Signatures of Fetal Hyperthermia.
Tonight's pubcast stories are brought to you by ReaShure Precision-Release Choline. ReaShure is the most researched encapsulated choline on the market today, consistently delivering results to your transition cows of higher peak milk, reduced metabolic disorders, and even in utero benefits to her calf, leading to growth and health improvements. Visit balchem.com to learn more.
Clay, what are some of the things the audience will want to pay particular attention to as they listen to this presentation?
Yes, I've really been fascinated by this research for a number of years now that started at the University of Florida. Dr. Laporta does a very nice job summarizing the short and long-term effects of in utero heat stress, really the last 6 weeks of gestation. And it's amazing the impacts that heat stress the last 6 weeks of gestation has, first of all, on the dam herself and, productivity in that succeeding lactation, basically a 5 kilo loss in milk yield, which is just amazing. But also how it affects both the calf that's born to that heat stress dam and the granddaughters born there. So it's she does a very nice job laying that out during this presentation. So they've really gone out to the third generation now to show productive impacts going out at least to the granddaughters now.
Yeah, very interesting. Thank you for that overview, Clay. And now let's go to the presentation.
Well, I would like to thank Clay for that introduction and Balchem for the invitation to speak today. And I want to thank Dr. Britt for this outstanding presentation on the key mechanisms of epigenetics. And what I'm going to do today is I'm going to expand on one of the examples that he gave and try to convince you and show you how heat stress during a relatively short period of time can really exert a phenotypic effect that lasts for multiple generations, but also a molecular signature, an epigenetic signature that can also remain for multiple lactations and multiple generations. All right, so this is going to be the outline of the presentation. I'm going to give you a little bit of background on heat stress, fetal programming, and the epigenome, and then we're going to dive right into the phenotypic signature, those short and long-term effects of in utero heat stress. And then we're going to move on to the molecular signatures on the mammary gland and the liver, and trying to uncover those genes that are shared between these two. So it's now news to this audience that heat stress is one of the largest challenges to the efficiency and productivity of dairy cattle. So on one hand, we have global temperatures rising all across the world, and on the other hand, we have modern dairy cattle that are high producing and they're generating a lot of metabolic heat production, and they're becoming more and more sensitive to even small changes in temperatures. So traditionally, the focus of heat stress research and the development of mitigating technologies have been focused on lactating cattle. And there's a good reason for doing that. If a cow is heat stressed, if a lactating cow is under heat stress, she will immediately drop milk production, dry matter intake, and she will become a manipid. So we'll definitely need to continue to cool our lactating cows. But heat stress does not discriminate, and it will really also take a toll and impact the physiology, productivity, and also welfare of dairy cattle in general, independent of the age, independent of the physiological status of the animal. And that's where I have spent a lot of my time researching on the heat stress effects on the calf, on the nulliparous heifer, and on the dry cow. And we're going to focus on the dry cow today. What's the impact of heat stress on the dry cows? This is sometimes overlooked, or at least paid less attention. But we have to think that during the dry period, the cow is undergoing, the mammary gland of the cow is undergoing two very important developmental events. Involution, that starts right after milk stasis, and that's where it's characterized by cell death. And at this point in time, we want cell death to get rid of those worn out cells that have been producing milk for the last 10 months. And then we move on to the redevelopment phase. As the cow approaches parturition with the hormones of pregnancy, those mammogenic hormones are going to stimulate cell proliferation and producing new cells that are going to start secreting milk as the cow calves. And so we know that heat stress really derails exposure of dry pregnant cows to heat stress, derails these cellular processes that are highly regulated, and consequently the cows are going to produce less milk in the next lactation. How much less milk? Well, on average, 5 kilograms per day in the entire lactation. And this is just a summary slide. This is just a summary slide showing 14 studies in different countries using different cooling systems, varying from just simply shade to more sophisticated cooling techniques with fans and soakers for dry cows. But the bottom line here, and this is just milk yield, so the bottom line here is that regardless of the country or regardless of the cooling method used, we can see that if cooling is not provided to dry cows under heat stress, and that's represented by the red columns, they're going to reduce milk yield by 5 kilograms, okay, regardless of the country and regardless of the cooling method used. Now, we also have a problem here because the dry cow is also pregnant at that time, and this is what Dr. Reed was alluding to. During this time, we have, I will say, 2 generations now under the spotlight, and so it is not only about the involuting mammary gland at this time point, but the dry period also coincides with the last trimester of gestation, which is a period of exponential growth of the fetus. And we now know that maternal heat stress during that time will lead to intrauterine hyperthermia, and that will result in smaller daughters that are born to those cows that are less able to survive in the herd, and they are going to produce less milk in their first lactation. So that's 2 years after they are born, or 2 years after that the initial insult was exerted on the mother. So this was the state of the art when I started studying this area of research, and I started asking some questions and looking at this from different angles. So what is the mechanistic linking the intrauterine environment with future productivity? What happens beyond that first lactation? Is it transient or is it multi-lactational? What happens beyond the first generation? Is only one generation affected or 2 generations? So I'm particularly interested in understanding how the fetal programming effects of in utero hyperthermia and how those result in lifelong implications. So the fetus is going to be extremely sensitive to the maternal influences in general, and those can be positive or negative. Clearly, heat stress is a negative influence on the dam and the offspring, and this happens at all stages of gestation. But if you think about the late gestation, we think that all the organs are there, the the offspring is just growing, the fetus is just growing, but there are still very important dynamic, this is a very important dynamic period of developmental plasticity in the developing offspring. And there's fast developing cellular events such as cell division, cell differentiation, the establishment of cell hierarchy as well, that are regulating those later stages of development and organ maturation. So in utero hyperthermia can really derail the developing trajectory of organs and systems, for example, the neuroendocrine system and the communication between the brain and the endocrine system, organ growth in general, the structure and function of the organ, overall metabolism, overall physiology of the offspring, and also the germline. This is what Dr. Britt was alluding to earlier. The fetal daughter is developing the oocytes at that time, so that's where the epigenome comes in, and we think that intrauterine hyperthermia is altering the epigenome. So the epigenome comes in as one of the potential mechanisms linking this intrauterine environment with postnatal performance. And as he alluded early, epigenetics simply means above or in addition to the traditional genetic basis of inheritance that we all know about. And it's not more than, in essence, it's a simple concept, but the biology is very complex. So it's not more than chemical changes that are going to alter the DNA physical structure without changing the sequence. So you have methyl groups that are going to physically bind to the DNA, or you have other factors that are going to bind to the tail of the histones, and that's going to alter the accessibility of the DNA to the machinery, and that will lead to the regulation of what genes are going to be turned on and off at a specific cell type or organ. And so the epigenome here is playing an important role in orchestrating this interplay between the environment and the genome, determining the phenotype that we see, what we measure, milk production, growth, and stuff like that. So there are three main mechanisms that are causing epigenetic changes. We have histone modifications, non-coding microRNAs, and DNA methylation. We and others focus mostly on DNA methylation because it's the one that we understand more, and it's more stable of the other types of mechanisms that we know. And if we think about the 101, if we have the promoter of a gene that has a methyl group on it, the gene is going to be repressed. The expression of that gene is going to be repressed. We now know that this is not always true, but this is kind of the basis of DNA methylation. So the way I like to think about this is environmental signals that are going to be received by the epigenome, recorded by the epigenome, remember across the cell generations and cell division, and revealed in an altered gene expression and cell function. Or in other words, a specific transcriptome of that cell or that tissue, that individual. But they also can be reversible. So this is where the epigenome comes as a key mechanism allowing for phenotypic plasticity within a fixed genotype. And this is what Dr. Reed was alluding to us earlier. So we're going to move on to look at the phenotypic signature of intrauterine hyperthermia, and we're going to classify that in the short and long-term lasting effects of heat stress. So one of the hallmarks of postnatal heat stress, of in utero heat stress, that I classified as short-term, and short-term I refer to pre-weaning, once that calf is born and during the pre-weaning period. One of the hallmarks is a reduction in gestation length. So those calves are going to be gestated on average 5 days less that those born to in utero cool dams. So there's less time for them to grow, and there's less time for the mammary development of the dam. They are also born smaller, and this small body weight is not only at birth, but it remains at weaning. On average, there's a 10% reduction in body weight, and also they are born smaller, and this reduction in size remains for at least 1 year of age. So we see that they have reduced hip height, reduced body length, reduced chest girth, and significant reductions in head circumference. And all of these are hallmarks of intrauterine growth restriction that we're more used to hearing in the human literature. But these are all hallmarks of in utero heat stress. Not only the body weight and the size of the animal, but also as a percentage of body weight, we see there's organ weight differences. We see alterations in immune organs. We see enlarged adrenal glands, which are the ones producing cortisol, and you might relate stress to cortisol. We see smaller ovaries at weaning in these heifers that are born to in utero heat stress dams. And we also see smaller mammary glands. And I'm a mammary physiologist, so this is very interesting to me. So we see this is an example of a mammary gland that we harvest at weaning. So after weaning, so this is 63 days of life. And you can see that the ones coming from in utero heat stress are quite small compared to the in utero cool ones. Not only small in length and size and weight, but when we dissect the tissues that are within the gland, we see that they have less fat pad and less parenchyma tissue. Okay, the fat pad is going to be supporting tissue that the parenchyma is going to grow into, so they have less room to grow and less parenchyma tissue to start off their life, their early life. When we look at inside those parenchyma, the parenchyma tissue, we look at the tissue microstructure, we see that they have fewer and smaller epithelial ductal structures. So in utero heat stress, those 46 days that the developing heifer is under heat stress in utero is going to limit normal mammary gland development during the developmental trajectory during this time. We see also impairments in innate and adaptive immunity. They have lower circulating immunoglobulins at birth and throughout the pre-weaning period, and they have reduced apparent efficiency of absorption, so they are less capable of absorbing immunoglobulins for colostrum. Even if you give them the same amount of immunoglobulins, they are absorbing less, on average 15% less. They have lower peripheral blood mononuclear cell proliferation, so overall a less robust immune response. So they have greater odds of having a health event, and they require more assistance from the personnel, from veterinarians, and they require more treatments during that pre-weaning period. So they are a lot of work. They also eat less. When we measure grain intake during that pre-weaning and post-weaning period, we see that particularly during those weeks approaching weaning, they eat on average 200 less grams of grain, and that's also during the week of weaning and the week after weaning. We see that effect. And also they have impairments in the way they thermoregulate. So they were under in utero hyperthermia, and once they are born, their body core body temperature is on average 0.2 degrees Celsius higher than those that were born to an in utero cool dam. And that's true for the entire pre-weaning period. So they are more susceptible in a way to heat stress once they are born. Now, to look at the long-term effects of heat stress, we did this retrospective analysis of 10 years worth of data from the University of Florida. We collected AFI records and we look at different lifetime events. So we were able to gather 400 dams that were either under heat stress or cooling during the entire dry period. And that's the first generation, a little bit different terminology to Dr. Britt here, but we call those F0 or the dam. And then we were able to follow 160 daughters, so we call those the F1 or daughters, and then 50 granddaughters or F2s in our data set. So having this large data set of pregnant dams, daughters and granddaughters, allowed us to look at the productive life and lifespan, milk loss, and ask the question if it was transient or if it was multi-lactational, and look at if it was multi-generational as well, and briefly at the financial implications of this. So this is the survival analysis for the F1s, so those daughters. And you can see here we have the survival percentage in the y-axis and the age of the animal in the x-axis. So you can see there's a trend here where the red line represented the heat-stressed daughters. It's always below the black line that represents the in utero cool daughters. So in utero heat stress is going to reduce survival to first calving, survival through the first, second, and third lactation, and overall productive life by 5 months, and the lifespan of the animal in general for almost 12 months. Now, those first lactation heifers that actually make it to lactation, these are the daughters again. This is milk production across the first 35 weeks in milk. We see the same trend. That red line is always below the black line, and that red line represents the heat-stressed daughters. So on average, for 35 weeks, they produce 2.2 kilos less compared to the cool daughters. That's their first lactation. Those that make it to the second lactation, we see the same trend. They produce, on average, 2.3 kilos less for the first 35 weeks in milk. And in the third lactation, we see the same trend. They are going to produce less milk. And this is milk yield, but we also see reductions in fat and protein yields as well for at least these 3 lactations that I show you. So there's this clear in utero programming of this less productive phenotype with lifelong consequences as I just show you.
Clay, last week, Dr. Britt talked about how methylation works, and this week, Dr. Laporta tells us that in utero heat stress can trigger methylation patterns. Can you summarize that for us, the relevance of this, and expound on why and how we can mitigate the effects? Can methyl supplementation help?
Yeah, it can. So, she does a very nice job explaining, first of all, how it's impacting mammary cell development in these animals, as well as a number of other things, ovary size of the fetus that's in there. There are a lot of impacts, both on the fetus that's inside this heat stress dam, and again, going out to the granddaughters at this point, right? Because when you have a pregnant dry cow, there really are 3 generations sitting there that you're impacting. You're impacting that dam herself, from a productivity standpoint in this succeeding lactation. You're impacting the fetus that's inside of her, but also you have to remember that if that fetus is a heifer calf sitting inside of the cow, that all of her oocytes are present. So you're affecting that 3rd generation through the oocytes that are being developed in that calf. So DNA methylation can play a huge role here. Again, turning genes off and on. And, certainly a methyl donor like choline could play a key role here. Of course, the first thing to remember is cool these dry cows, right? We want to try to mitigate the effects of heat stress as much as we can in late gestation. That's the first thing that to glean from the research. But certainly, having adequate supply of methyl donors in late gestation will help as well.
All right. Thank you for that explanation, Clay. And now back to the presentation.
So to answer the question if it's multi-generational, I think the answer to that is yes. This is the survival curve for the granddaughters. And we see again the same trend, the red line representing the granddaughters, okay? It's always below the black line representing the granddaughters. And clearly we have fewer animals here. And so this is exciting to see that these animals are also producing less milk. This is a part of what Dr. Reed showed in his presentation. But on average, in the first lactation, those granddaughters, and this is 4 or 5 years down the road, where the initial hystress insult on the pregnant dam occurred, they produce 1.3 kilograms less per day. We also look at the second and third lactation. Clearly, we have fewer animals by then, so we don't even show that. But the trend, numerical trend remains. So this late gestation heat stress exerts carryover effects on at least 2 generations or 3 generations, depending how you want to see it. And this is 3 or 4 years down the road when the initial heat stress insult occurred. How much is this costing the dairy industry? I think we have a good estimation of the financial loss in lactating cows at $1.5 billion by Sampier in 2003, and also for dry cows by Ferreira in 2016 for estimated $800 million for the loss in milk of that dry cow under heat stress across the U.S. But I think we would be underestimating these estimates if we don't consider the losses in the progeny of that dry pregnant cow, of the offspring of that dry pregnant cow. So we wanted to estimate that, and we did that using the milk yield and survival data that I just showed, and assuming that no cows in the U.S., no dry cows in the U.S. were cool. So this is showing 25 states across the U.S. and the $1 million loss in the x-axis. And so what we did here, we split it up in heifer rearing costs, that increase in heifer rearing cost in those animals, the decrease in productive life, and the decrease in milk production of those by state. So when we look at collectively, we see that across the U.S., those losses can be up to $600 million. And this is just looking at the daughters, okay? So we see here the tri-state here, Indiana, Michigan, and Idaho. You can have, I think it's $16 million and $18 million losses if dry cows are not cool. So if you think, if we combine these with the losses of the dry dam, the total annual loss due to late gestation heat stress can climb up to $1.4 billion, and that's comparable to the estimates for lactating cows. So not saying that this is more or less important, but if you think about it, can really have a huge economic impact if we consider the losses in the offspring. And this is not even considered the granddaughters or the nulliparous heifers under heat stress. All right, so I show you all the phenotypic effects of in utero heat stress, and we're going to now move to the molecular signature of in utero heat stress. We're going to focus on the mammary gland and the liver. So before we move on, I want to just remind you that when we think about milk production during lactation, we put this as a function of the number of mammary epithelial cells that are present within the mammary gland, and that's a function of the cell growth, a ratio of the cell growth and cell death, and also the mammary epithelial cell activity or metabolism, and that can be related to the genes and proteins that those cells are expressing and how they are regulated. So what we wanted to look was the carryover effects of in utero heat stress on mammary development and function, so tackling both sides of the equation. So what we did here, we went in 2 years later and we took a mammary biopsy on the first lactation, on those daughters that were under in utero heat stress or in utero cooling when they were being gestated. Okay, so 2 years after when they start their first lactation, we took a biopsy at 21 and 42 days in milk. And what we did with that, we put it on a slide and we look it under the microscope. We do many different stainings. What we see here is the first column is the in utero cool mammary glands of those daughters and in utero heat stress daughters. In the second column, AL refers to the alveoli, which is the synthetic unit of the mammary gland, and is composed of a single layer of mammary epithelial cells. And remember that this is a two-dimensional image, but in reality, those are 3D kind of like balls of a single layer of mammary epithelial cells. And what we did is we look closer into, I think you can clearly see how the alveoli of the in utero heat stress are shrinked, are smaller. So when we look closer into that tissue, we see that the lumen, the luminal alveolar area of those alveoli, is quite smaller. It's 46% smaller. And when we count the number of mammary epithelial cells forming that alveoli, we see that there are significantly fewer mammary epithelial cells composing those alveoli. Remember, each individual cell is responsible for making milk, right? So fewer cells, fewer milk synthetic capacity. We also see that these cells have lower proliferative capacity, so they were proliferating less, and there's more connective tissue. We see here in blue in the second column, you see a lot more blue, and the stroma is important to support the secretory tissue, but during lactation, you want to maximize the epithelial structure, the secretory tissue, and minimize the stroma tissue. So this is a little model that we put together, but the overall conclusion here is that we see compromised synthetic capacity and milk storage capacity in the in utero heat stress daughter's mammary gland. And when we combine these are two undesirable characteristics during lactation, and we think that this might be contributing to the reduced lactation performance that we see in these daughters. Now, with the same samples and the same model, we wanted to look at the DNA methylation pattern, asking the question if these alterations in DNA methylation, are these triggered by in utero heat stress? And can we explain the phenotypic outcomes with these? So we did reduce representation by sulfate sequencing to answer this question. We found 523 differentially methylated cytosines in the mammary glands of the in utero heat stress daughters, and those were associated with 135 differentially methylated genes. So when we look at what genes, we see that most of them were involved in stress response, mammary development, and milk synthesis. There's a handful here, hypomethylated genes in utero heat stress and hypermethylated genes in utero heat stress. We see tyrosine kinases involved in cell survival, cell migration. We see protein kinases. We see protein ligase involved in cell-to-cell communication. So very important pathways affected there. But the important genes. But when we look at the pathways that these genes were involved with, we see that these differentially methylated genes were associated with more fundamental cellular processes such as genomic stability, cell survival, transcription, translation. So we kind of asked the question, are these differentially methylated genes in the mammary gland that we're seeing, are those unique to the mammary gland? Or are there common patterns of DNA methylation that are induced by in utero heat stress, regardless of sex, of age, or tissue type? So we compared the data set that we had, those 135 genes in the mammary gland, with differentially methylated genes in the liver of bull calves at birth, so completely different sex, age, and tissue type. And we look at if there were any common patterns there. And we were happy to see that we have 50 genes that were the same in both tissue types that were shared between the two data sets. So we see 20 of those that code for ribosomal RNA. So we know how important that is in translating the mRNA into protein. And so in utero heat stress seems to be affecting the protein synthetic machinery, and that can have a huge impact on protein synthesis. Mitochondrial function, ATP synthesis, we see a few genes involved in that. We know how important energy is for lactation and for growth, so we see that as well. DNA repair, genomic integrity, cell adhesion and migration. Intracellular signaling, we see phospholipase C, you might have heard about it at some point in your life. That's a very important intracellular signaling pathway affecting multiple other functions, right? So this was exciting, but we had another question, and that question was, are these differentially methylated genes that we're seeing uniquely methylated in response to in utero heat stress or heat stress in general? So we compare in utero prenatal heat stress with heat stress postnatally. So here we use the same tissue, so we use the data set that we have those bull calves born to in utero heat stress or in utero cool dams, and we compare that data set to the differentially methylated genes that we found in the liver of calves that were heat stress or cool postnatally, so a very different developmental stage there. And again, we find 50 genes, and this 50 is just a coincidence, it's not the same 50 genes. So we see hepatocyte proliferation, not surprising. Wind signaling pathway, very important pathway. Ask Dr. Hansen, he knows all about it. Angiogenesis, cell signaling again. We see a protein kinase here that phosphorylates a lot of proteins downstream and affects different functions. So we were excited to see that there are indeed common patterns of DNA methylation that are induced by both prenatal but also postnatal heat stress. So we wonder, are these environmentally driven epigenetic effects? So regardless of the tissue, the gender, and the stage of life, heat stress is impacting specific genes. So here we found five genes when we compare all data sets. And that was pretty exciting, and we're definitely looking more into these genes and as we speak. But some of them are quite interesting. We see COX-1, it's a very conserved pathway in mammalian development. It's a DNA-binding protein that regulates cell morphogenesis, differentiation, cell cycle progression. So we also see NOT4, which is very conserved signaling pathways as well, involved in cell differentiation, proliferation, apoptosis, and also branching morphogenesis, which is particularly important in the mammary gland. We also see, again, this phospholipase C that I mentioned earlier, involved in intracellular signaling, and it's using calcium as a cofactor to catalyze the formation of diacylglycerol and IP3, and that's involved in the transduction of extracellular signaling. So heat stress, whether it's prenatal, postnatal, in whatever tissue we're looking at least for now, we see these consistent patterns of genes that are methylated by that. So in summary, in utero heat stress induced fetal programming of the offspring, we see these very marked phenotypic effects in growth, organ development, immune function, survival, longevity, and milk yield. And we see that in terms of milk yield, when we look more into the mammary gland, we see that in utero heat stress is derailing the normal developmental trajectory of the mammary gland. We see smaller alveoli, which are the secretory units of the mammary gland, that have fewer mammary epithelial cells, which are the cells that are making the milk, so you have less to start with, so you have a problem. And those have less synthetic capacity. Now, we see that in utero heat stress triggers these distinctive but also unique methylation patterns that are organ-specific in terms of the mammary gland and liver, we see those that are unique to the mammary gland, and we see common differentially methylated genes in the liver and the mammary gland. And we're postulating these environmentally driven epigenetic changes to specific genes, and definitely doing more work in that area. We also see that these phenotypes that we discuss persist until at least the second generation, or the granddaughters, if you will. So there are multi-generational effects. I cannot say if those are going to be transgenerational, because we would have to look at the third generation, so the F3, to really be sure that those are transgenerational. But we do see that these F2s, these granddaughters, survive less and produce less milk 3-4 years down the road. So some final remarks here. I want you to think about the importance of the dry period. And why is that? Is because there are 2 programming effects, or maybe 3, I should say. One is the mammary development of the dam, and heat stress impacting her ability to produce milk in the next lactation. The second one being the fetal development of the daughter. We often don't think about the early life developmental progression of the mammary gland, and we start thinking about that at puberty. But we can clearly see that if we give them a bad start, they are gonna, produce less milk. So I like to think about this as an opportunity for the implementation of management interventions. Here we're discussing cooling or heat abatement in this case, but perhaps nutritional intervention, of a combination of both of those that can have really long-term impacts. And it's a really relatively short period of time that we can generate a long-lasting impact. So with that, I would like to thank Balchem for the invitation to speak today, my former and current institutions for their financial support across the years, and USDA NIFA for funding, Jeff Dahl, collaborator at the University of Florida, and students and postdocs, but particularly Amy Skibble, Veronique Wallet, 2 postdocs that did most of the work, and Bethany Dadosen, a PhD student as well.
Tonight's last call question is brought to you by NitroSure Precision-Release Nitrogen. NitroSure delivers a complete TMR for the rumen microbiome, helping you feed the microbes that feed your cows. To learn more about maximizing microbial protein output while reducing your carbon footprint, visit balchem.com/nitrosure.
Clay, any thoughts you'd like to leave the audience as we close this one out?
Yeah. Again, cool the dry cows. And it's not just those last 3 weeks before calving. If you look at all this research that's been published, mainly out of the University of Florida, you have to cool these cows the last 6 weeks of gestation. There are some things happening very early on during the dry period that we can't make up for just by cooling dry cows the last 3 weeks of gestation. So we need to really emphasize cooling dry cows the full dry period. That'd be my first takeaway. And then the second one is to really think about methyl donor supplementation, particularly choline supplementation. And we really haven't looked at this specifically in a heat stress situation at this point, but we certainly have indications that that choline supplementation of these dams the last 3 weeks pre-calving has very positive impacts on the calves born to those dams.
Well, thank you, Clay. If you'd like to view Dr. Laporta's full webinar and slides, go to balchem.com/real science and scroll down to April 27th, 2022. The title you'll see there is the 2022 Tri-State Dairy Nutrition Pre-Conference Symposia, Exploring In Utero Influences on Transgenerational Performance. Click the link, and all 4 of the presentations will be there for you to view and to click on. Clay, as always, it's been a real treat having you here now in the well-worn bar stool here at the Exchange. To our loyal listeners, be sure to look for our next podcast in the series next week, when we will be featuring Dr. Eric Capio from Balchem Corporation. His talk is titled The Growing Importance of Choline in prenatal human nutrition. As always, thank you for joining us here tonight for the conversation. We hope you learned something. We hope you had some fun. And we hope to see you next time here at the Real Science Exchange, where it's always happy hour and you're always among friends.
We'd love to hear your comments or ideas for topics and guests, so please reach out via email to anh.marketing@balchem.com with any suggestions, and we'll work hard to add them to the schedule. Don't forget to leave a 5-star rating on your way out. You can request your Real Science Exchange t-shirt in just a few easy steps. Just like or subscribe to the Real Science Exchange and send us a screenshot along with your address and t-shirt size to anh.marketing@balchem.com. Balchem's Real Science Lecture Series of webinars continues with ruminant-focused topics on the first Tuesday of every month, monogastric-focused topics on the second Tuesday of each month, and quarterly topics for the companion animal segment. Visit balchem.com/realscience to see the latest schedule and to register for upcoming webinars.